Hello everyone. Today we'll be discussing the contents of week 9, followed by some questions pertaining uh, to the current week. So, in the previous lectures, we have looked at the anatomy, the structure of different uh, portions of the human body, the muscles that supply to those areas, the bones that are present in those areas. We've looked at the concept of elasticity and the relation between in viscosity and elasticity for hard tissues, different models that tell us how to analyze viscoelasticity. This week we'll be studying the mechanics of soft tissues. Now, by soft tissues, it's essentially uh, ligaments, tendons, cartilage, etc. So, we'll look at different constituents of the tissues, which are mostly matrix proteins. These are different matrix proteins such as collagen, elastin, and proteoglycans, which make up the bulk of, apart from the cellular portion of the muscle or tendon or ligament. These are the proteins that are present in the extracellular matrix, which is outside of the cell, and this is what uh, provides the structure to the tendon or ligament. Majority of what provides. Okay, so coming to the tissue constituents, uh, primarily there are four things that you need to know: collagen, elastin, proteoglycans, and glycoproteins. Now, collagen provides toughness and flexibility. This is what provides the skeleton of sorts to the tendon or to a ligament, etc. It is also flexible. It has a particular structure which we'll be seeing in the subsequent slides, because of which it is very flexible, and extension of the tendon or ligament is possible. Next is elastin, which provides strength and stretchability. Elastin is a protein that is very elastic, and therefore it is essential for maintaining the stretchability of any structure that it is present in. There is also proteoglycans, which provide firmness to the to a tendon or a ligament, in addition to collagen. And there are certain other proteins known as glycoproteins, which are present on the cell surface. Because they are present in the cell surface, they are essentially outside of the cell. These proteins might be embedded in the cellular membrane, but these are the proteins which uh, mediate the adhesion of the extracellular matrix and the cell. So the you need to hold together the cell plus the matrix, so that the tissue does not disintegrate, right? So these glycoproteins are the ones which mediate the adhesion, and therefore these are known as cell adhesion proteins uh, of the cell to the matrix. uh, some of these proteins are laminins, fibronectins, pollicans, etc. There are many such examples. Coming to the first one, let's see collagen in more detail. It is one of the most abundant proteins in the human body. It makes up to one third of the total body protein. It's present in various tissues, it's present in bones, it's also present in the skin itself, etc. It's present in muscles. Therefore, it is a very essential part of the human body. Uh, Genetically, there is a so if you look at the molecular biology of it, there is not one collagen molecule, there are multiple collagen types, and each of these collagen types are encoded by various genes. Just an extra thought. So these are the basic structural element for soft and hard tissues in animals. This is not only true for humans, this is also true for other animals, and they are present in bones, muscles, even skin, ligaments, tendons. This is just a short list. There are also present in other portions of the human body, but this is majority of what makes up the collagen bulk in a human body. So the structure of collagen is a triple helix. As you can see, the essentially three collagen fibers wrap around each other uh, to make a triple helix, and this is one collagen fiber will be very weak. These three collagen fibers, owing to their structure, have very high load bearing capacity. So it has a triple helical structure. It also it gives mechanical integrity and strength to the tissue it is present in, and it is present in different forms. It can be a load carrying element in uh, some of the examples are blood vessels, skin, tendon, and bone. It The other types are type 1, which are present in ligaments, tendons, and bones. Type 2, which is mainly found in cartilage. It's very difficult to break up. Okay, okay. I'll come to it later. The collagen properties are mainly dictated by its structure, its composition. Because of its triple helical structure, it is very strong. If you were to compare a single strand of the, if you take a single strand from the triple helix and compare its strength to the complete triple helical structure, you'll find that the difference in the strength and the resistance to mechanical stress is very different, is very high in the triple helix. This is a scanning electron microscope image, uh, which shows that there is a characteristic waviness in the image, as you can see here by the red line. So it's like this. It's not a straight fiber, or it's not like, it might be like this. It might be very it might have uh, very small crimp, but it has a waviness to it nonetheless. What happens during a load bearing or under tension is that this crimp or this waviness gets straightened out, and depending on what stage it is in, the biomechanical properties of the collagen tissue will be different. Also, collagen is one of the proteins which is relatively resistant to enzymatic activation. Uh, Therefore, it's a very long lasting protein. There are enzymes that would degrade it, but they are not synthesized in normal conditions. As I don't think they are synthesized in the human body. These enzymes are known as collagenases. So, any it ases means something that is acting on an enzyme that is responsible for breakdown of something, and collagen uh, uh, is the protein it's acting on. Okay. So if you look at the stress strain curves for, so one more thing is, this triple helix is essentially known as tropocollagen, and multiple such tropocollagen form to make microfibrils, which can then form fibrils, which can then go on to form fascicles and other higher structures. If you look at the collagen properties, the stress strain curves for tropocollagen, collagen fibers, and tendon is very different. This is the blue dots mark the stress strain values for the tendon. The green is for collagen, and the red is for tropocollagen. So if you want to compute the modulus is essentially stress over strain. it is essentially the slope of this graph that will tell you about the modulus uh, of the item or of the object. So if we look at the modulus of compare the modulus of tropocollagen versus collagen. By collagen it means a single fiber of collagen, whereas tropocollagen is the triple helix structure. uh, and this is a single collagen fibril. This is a triple helix, and this is the tendon. Now, as you can see, triple helix has the capacity to handle, since the slope for the triple helix is higher, it the Young's modulus for the triple helix is more than triple helix is more than single collagen fibril. However, the tendon has multiple such tropocollagen or triple helical structures. The the modulus of the tendon is slightly different than, uh, is not a linearly elastic, or does not have a linear modulus. It has varying modulus. We'll look at that in detail in the later slides. For now, essentially, the modulus of tropocollagen is about 3 times greater than that of the single fibril. Let's look at elastin. This is the this is the protein that imparts elasticity to the tissues, and it's abundant in large elastic blood vessels such as aorta. It's present in lungs, skin, bladder, etc. Essentially, compartments in the human body where there is a need for uh, uh, elasticity and for strength also. It functions to impart elasticity and resilience to the tissues. In arteries, it also functions as a medium for pressure wave propagation to help blood flow. So there is the systolic diastolic pressure. Elastin aids in that. uh, The elastin composition is essentially tropoelastin molecules cross-linked by lysine. So tropoelastin is a precursor for uh, uh, elastin, and these tropoelastin molecules have lysine residues on them. The cross-link between these lysine residues leads to formation of the final elastin uh, polymer. Now, if you look at the stress strain curve for elastin, you can see that if this is uh, this there is a gap between the work there is a gap between the way increase in stress and then the final decrease in the strain, which is essentially hysteresis. So, and hysteresis is usually present in viscoelastic uh, viscoelastic materials. So that implies elastin is a viscoelastic material. Okay, and so if you were to fix any tissue, it essentially arrests the tissue at the state at which it was fixed and kills it. So the stress strain that the tissue can tolerate will be different than a live tissue. Looking at proteoglycans, these proteoglycans are essentially large molecules of central core protein with covalently attached glucosaminoglycan side chains. So this red thing that you see is the core protein. The and these have and there are multiple such structures connected to each other to form the final protein. These are very large molecules. There are multiple such subunits present in each of the protein, final protein, and uh, these blue things that you see are the glucosaminoglycans. Essentially, it forms a brush-like pattern, and in most cases, it forms a mucosal layer over a tissue or inside a tissue. Lubricating layer, might not be a mucosal layer. Glucosaminoglycans contain sugar, therefore they have negative electrostatic charge on the protein core. Glucosaminoglycans are essentially sugar molecules. So because of this, they help in water retention, and therefore they help in maintaining the turgidity of a tissue. If you look at the compressive modulus of glucosaminoglycans, with the amount of glucosaminoglycans, the compressive modulus increases. It might not be linear. This is a linear fit, but it's not a linear relationship. But in general, the compressive modulus of elasticity of a protein in which glucosaminoglycans are present, uh, the compressive modulus depends on the amount of glucosaminoglycans. More the amount, more the compressive stress. Also, uh, in if you increase the cartilage depth, if you go inside the cartilage, the glucosaminoglycan concentration increases, and therefore the tissue compressive stiffness also increases. Also, stiffness, compliance, elasticity might be used interchangeably in the slides, just to note. Okay, we looked at tissue constituents. Let's look at where these are present. So these are usually present in, for our purposes, we will be looking at cartilage, ligament, tendons. These are present in other places as well, as we saw. In So cartilage are of uh, various types. The first type is hyaline, which is the most common one. It is present on the surface of uh, on the articulating surface of the bones. If this is one bone and there is something that is attaching here, it will be present at the junction of these two bones. One of the examples is the ventral surface of the ribs. The another type of cartilage is fibrocartilage. It is a it acts as a load bearing component, and therefore it is present in intervertebral discs. Uh, the third type is elastic cartilage, which is flexible and is usually present in external ear and eustachian tube. If you see the structure of these, the structure also varies. You can see that the hyaline cartilage have uh, very little fibrous material in it, whereas fibrocartilage, as you can see, have these fibers in them, which is essential because they have load bearing function. And elastic cartilage have these networks in them, is which aids to the flexible component of these tissues. Coming to articular cartilage or the hyaline cartilage, these function in transmitting forces across joints. They essentially distribute the forces such that the stress concentrations are minimized. They also provide a smooth surface for the gliding of the joint surfaces. Like if there is movement, they are responsible for uh, ensuring that there is no wear and tear of the bone itself. The structure is not homogeneous, it's heterogeneous, and the material properties change with depth. As we saw, the amount of glucosaminoglycans also changes, and that also contributes to changing the stiffness of the cartilage, therefore changing the material properties. Now, if you look at the structure, if this is the bone, if this is the surface of the bone, there is some calcified cartilage just near to the bone, and it is essentially the deep zone. There is a middle zone which forms the majority portion, which is packed but not as packed as the calcified cartilage, and there is a superficial zone which is very loosely packed, and therefore depending on where you are, the material properties will change. And there can be different components. There can be hyaluronic acid, there can be type 2 collagen, it can be proteins containing glucosaminoglycans, uh, they and there will be of course water. Coming to ligaments, these are very tough bands of fibrous connective tissue. These essentially connect one bone to the other bone in joints. They are responsible for providing stability by limiting excess movement, therefore prevent dislocation in most cases. They have some elasticity, but uh, they comparatively they have some elasticity owing to the presence of elastin, which is more than that of tendons. Coming to tendons, these are these are also fibrous connective tissues. These are flexible, and these are responsible for attaching muscles to bones. These can withstand very high tension and stress because they are responsible for force propagation. This facilitates the movement by transmitting the force generated by contraction of muscles to the bones. Uh, Compared to ligaments, where ligaments are responsible for holding the bones in place and preventing excess movement, tendons actually facilitate movement by force transmission or force propagation. Now, if you look at the organization of fibers from a top down approach, the tendons follow a structural hierarchy where fibers are bundled to form microfibrils, subfibrils, and fibrils. And ligaments also have a similar hierarchical range. So, if you look at tendons have fascicles. These fascicles further have fibrils, and these fibrils further have subfibrils, microfibrils, and tropocollagen. Note that these tropocollagens are in nanometer range, whereas tendons are in micron or millimeter range usually. So you can appreciate the arrangement in these different stages. If you were to look at the biomechanics of tendon, in general, the bundles of collagen fibers in tendon are more parallel in the collagen fibers are more parallel in the tendon than in a ligament. This essentially adds to the elasticity of the ligaments. If they were crisscross, or if they were like this instead of being like this, the effective elasticity of the material will come down. Okay. Let's look at the stress strain relation in tendons. As we saw earlier, that the stress strain relation in tendons is not linear, it's non-linear. um, in so let's look at it. In general, the elastic behavior of any material is represented by either forced and the deformation caused by the force, and stress strain relation, either one of those graphs. In whatever in and whatever you are looking at will give you what give you a parameter which you can further uh, which you can further which can be further used to classify what kind of material it is. So if you are looking in the force deformation condition, would sorry you would either compute tendon stiffness or compliance, which is 1 over the stiffness, uh, or you can compute the Young's modulus, which will give you the an idea of the elastic behavior of the material. Here, because it's a non-uniform setup, the Young's modulus is also known as tangent modulus of elasticity or elastic modulus, which is essentially the slope of a stress strain curve. If your stress strain curve looks something like this, to compute your modulus, you need to draw a tangent, take the values there, the stress and the strain values, and you will get an idea of the elastic modulus. Okay. So essentially, the Young's modulus for a tendon can be regarded as the stress required uh, to make a delta uh, make uh, a strain of 1, essentially, because the strain is a dimensionless quantity. So if you it you can also represent Young's modulus by as a product of the tendon stiffness and the ratio of the original length to the cross sectional area. Both of these will give you the same value. uh, in some joint configurations, because of the crimp, the tendon may be slack and therefore it does not produce a resistive force when elongated. The thing is, this is usually not true, because in any configuration there will be some resistive force uh, in the body part, whichever you are looking at. Okay. So if you look at the stress strain curve for a tendon, you can see that it's a sigmoidal curve, it's not a linear curve. And the slope of the stress strain curve depends on the tendon elongation. What condition the tendon is present in gives an idea of where the stress lies in these areas. So there are essentially three areas. One is a toe region, which is here. This is the region where upon where the change in the stress and strain is not linear, where the slope increases essentially. Then there is a portion here where the slope is constant. This is a region where there is microscopic failure, where all of the fibers have become stretched. There is microscopic failure starting to occur, and then there is failure or rupture of the tendon. So three regions: initial toe region, linear region, and failure region. Three different regions. if you look at the ranges for strains in these regions, the tendon can be strained to approximately a 2% on an average, with a range of 1.5 to 4% uh, in the toe region. So initially, the at the starting point, the fibers are crimped, and as they straighten out, they enter the linear region, and they might, some of these fibers, because they are beyond the capacity of their strain, they might enter microscopic failure, following which they might go into macroscopic failure. But in general, the range in this toe region, the tendon is strained to a, on an average of 2%. The only exception is the human psoas tendon, which goes from L1 to L4 vertebrae, essentially lumbar vertebrae, which inserts into the lumbar, originates from the lumbar vertebrae and inserts into the femur. It's inserts near the knee joint. It's a very large muscle, and this can, the tendon that is attached to the psoas muscle can extend to 10% strain. So under these, as I said, under these low strain conditions, collagen fibers straighten and lose their crimp pattern. But the fiber bundles themselves are not stretched yet. They only stretch after this toe region. And the modulus of elasticity increases because the slope increases until it reaches a constant value at the start of the linear region. And on an average, the slope that you get from the stress deformation curve at this in this area is called as the toe region modulus. Now, why there are a couple of hypotheses that talk about why there is a non-linearity in the elasticity in the toe region. The first model assumes that the consecutive recruitment of initial fibers, collagen fibers, have varying degrees of crimp. Essentially, there is a heterogeneity in the crimp pattern. Some of the fibers might have a crimp like this, some of them might have crimp like this, whereas some of them might have a larger crimp. And since there are fibers in different conditions, there is differential recruitment of the collagen fibers. This one might straighten out faster than this one, which might straighten out faster than this one, or the other way around. So essentially, with increasing tensile deformation, new load-bearing fibrils jump into action and resist the deformation. And the non-linearity arises because there are different number of fibrils in different states, and increasing number of fibrils contribute to the tendon resistance, thus increasing the modulus, and therefore you see a change in increase in the slope. And this model therefore explains how a large number of linear elements can of different resting lengths can produce a nonlinear force deformation curve. In this graph, the assumption is that there are three different springs, and in three different stages of deformation, and depending on that, they have different contributions to the stress strain relation. There is one more model which is known as the hinge model of tendon crimp, flexible hinge model of the tendon crimp, which assumes that the crimped collagen fibers are lengthened until they become straight, similar to stretching a helical spring until it becomes a straight wire. Uh, and the stiffness increases from a raw value for a spring, which is to a much larger value for a straight one. As we saw earlier, the linear region begins around 2% of the strain, which is essentially here. In the linear region, the tendon elongates. Tendon elongation results in stretching of pre-aligned collagen fibers, which are parallel. These fibers are parallel, and therefore the stiffness is indicative of what the collagen fiber stiffness is, or the inherent material stiffness. After it, in the linear region, which is essentially the slope of the, not a tangential slope, rather the slope of the linear region, is called as the linear region modulus. And if a tendon is stretched beyond 4% of its original length, there it enters into microscopic failure area. And except for the psoas tendon, which can tolerate up to, for which the toe region is 10%. And if you were to compare the values of uh, different stresses that these regions can fall under, the linear region starts in at around 50 to 30 megapascals, and failure region starts at 70 to 100 megapascals, and the Young's modulus in the linear region is approximately zero point 0.6 to 1.7 gigapascals. Now, how do you compute the tendon elongation? Essentially, the tendon length at zero strain needs to be computed first to determine how much elongation is there. And during in vivo measurements, because there are already forces acting, because no one is ever at complete rest, there are already forces acting in the during the measurements, which is usually imaging, various imaging techniques, mostly ultrasound. uh, because of this, it's very difficult to compute the elongation because you don't know the initial length. This can be overcome by using some uh, sorry, one sec. This can be overcome by using some invasive techniques such as tendon force transducer implants, uh, in the which are implanted into the near the tendon you are uh, looking to study. However, these are very invasive and usually not ethically not allowed to perform in various in in humans in general. Most of these measurements are therefore performed in laboratory animals, and therefore the comparison between human scales and the animals lab animal scale is very different, and therefore it is very difficult to get appropriate measurements for the for humans. So, and also one more problem that arises is because the strain pattern, because one is the zero strain length is not known. The other problem is that the strain pattern in some tendons are not uniform along their lengths, and uh, they also might exhibit stress shielded areas, or even areas that are subjected to compressive loading, especially at the bone junction. Because of this non-uniformity, there is very it it's very difficult to measure on human subjects the values for the stress strain relations in linear region and other regions in the graph. Now let's look at various forces that are present in the tendon. Um, in general, tendon forces can be very large, and because of some damping, the final force that is produced or that functions is that is at that is essentially the effect might be smaller than the force generated, because the moments are very small. The forces are very large compared to what it the surrounding environment will feel. The forces in the tendon are very large compared to the environment. For example, in flexor digitorum superficialis muscle tendon, the force that is generated is 5 times higher than the fingertip contact. Also, during typing, whatever forces are generated is 7 times the contact forces, which are essentially the effector forces. Are the effector forces times 7, the forces that are used to generate the effector forces are 7 times the effector forces themselves. In Achilles tendon, the forces produced can be huge when compared to the body weight. And this is sorry for the delay. Okay. Um. Okay. So Achilles tendon forces are huge when compared to the body weight. So it's around 12.5 times the body weight. In one of the pioneering studies in studying the studies for looking at the Achilles tendon forces was done by P.V. Comey, where they introduced a buckled force transformer in their own Achilles tendon and measured the forces, wherein they found that it's around 12 times the forces produced are around 12 times the body weight. And as we saw in using multiple examples, tendons can sustain and transmit huge amounts of forces. And essentially, a tendon with a cross sectional area of 1 square centimeter can produce uh, 10 kilonewton, can support 10 kilonewtons of load. E- So the stress will be 10. This, which is a huge amount of stress that the tendon is uh, supporting, is feeling. And these are uh, in a muscle, the stresses are around in the range of 25 newton per meter square centimeter. So however, these are daily activities. During various sports, the tendon forces can be huge. For example, during weightlifting, patellar tendon, essentially near the knee joint, the which can feels around 7.5 times, feels forces which are around 7.5 times the body weight, which is a very large force. Now let's look at aponeurosis and what are the differences between tendons and aponeurosis. Aponeurosis are essentially internal tendons, and they in general they gradually increase in thickness as they go towards external tendon. So This is the muscle. This is the internal tendon, whereas this is the external tendon or the tendon. Aponeurosis can transmit a large forces closer to the external tendon. However, there are no differences in the mechanical properties as observed in various animal experiments. There is no human data because of ethical reasons. However, between the internal and external tendons, the mechanical properties are almost the same. One more property is that one more assumption is that tendons and aponeurosis are both in series. However, there is no real human data. This is an assumption because of how they are structured. And this tendon stresses or tendon excursions can be huge. In triceps surae, it can go as high as 7% during running, which is not likely in the toe region. So, happens, whatever occurs in during daily activities usually lie in the toe region, whereas competitive sports or very high intensity sports are usually in the linear region. Aponeurosis and tendon strains vary based on muscle group and function. However, there are no clear pattern. There is no group function relations or strain function relations. So, tendons also have a limit. Uh, the forcible stretching of tendons is resisted essentially by three sources: uncrimping of fibers, fiber deformation, and tendon matrix resistance. And these three functions, these three sources can be modeled differently. Uncrimping can be modeled as an exponential function. The fiber deformation is considered to be linear, and uh, the third component, which is the tendon matrix component, is more of a qualitative thing because it depends on the components of the matrix, whether the matrix is compressible, incompressible. And one So therefore, bec- uh, in a lot of conditions, the z dimension is it's only two dimensions that are considered for modeling. The third dimension is ignored, which leads to various inaccuracies in the prediction. Because there is one exponential, one linear, and there are some inaccuracies, it is safe to assume that it's a viscoelastic setup. Again, another uh, property is the other factors that control the mechanical properties of the tendon are the length, cross sectional area, and the tissue property, the composition of the tissue. The depending There are other factors that control the tendon properties. One of them is gender. In general, women have lower stiffness but higher maximum strain, lower stiffness and modulus and higher maximum strain as compared to men. Age is another one of the factors that contribute to tendon properties. uh, Essentially, with age, stiffness and modulus go down, and however, the maximal strain also goes down. Hysteresis increases. uh, Exercise in general can help in mitigating some of these effects, but there will be some decay in the stiffness and modulus and maximal strain with age. With exercise, these the deterioration will be less as compared to without exercise, but there will still be some decay. the um, the chances of rupture and microscopic failure is also more with age. Disuse of muscles is also something that leads to decrease in the stiffness of the tendon. This usually happens in bed rest where there is some illness or some injury. In uh, in case of spinal cord injuries, which leads to bed rest, there is a atrophy of the tendon tissue itself. The Therefore the stiffness reduction might be irreversible in case of spinal cord injury because of various neurological issues then also. In During exercise, the tendon response is very specific. Low load resistance training may increase the maximal strain but no change in the stiffness of the modulus, whereas high load resistant training increases the stiffness and the modulus. It also increases the rate of force generation or force development. Just a second. Let's look at the tendon interaction with surrounding tissues. So, in general, there is mechanical interaction of tendons with other tendons. In very small areas, there might be multiple tendons present, and these mechanical interactions are known as intertendinous shear force and lateral force transfer. uh, when many muscles connect to a common tendon, there are different muscle displacements, and there will be some shear and therefore some force transfer occurring between these tendons. For one of the examples is during plantar flexion, the medial gastrocnemius and soleus aponeurosis deform to deflate levels. Excuse me. As a result, there might be sliding between the layers of the connective tissue that are parallel to the acting forces, and there will be some shear between these two because of the sliding. In general, force transmission in the tendons is along the axis, but there might be shear forces between the adjacent aponeurosis. One specific case of introtendinous transfer or tendon-tendon transfer occurs in hand, and it results in a specific phenomenon known as finger enslaving, where it is not possible to move. It's it's very difficult to move one finger independent of the other fingers. While moving one finger, other fingers might show some movement. The intent is to move, say, the index finger, but because the finger is moving, the ring finger, the middle finger, and the little finger also move. Thumb usually is not privy to finger enslaving. It's for digits 2, 3, 4, and 5. So, tendon interconnection are one of the reasons for finger is enslaving. The muscles of the hand can be divided into two groups, extrinsic and intrinsic. Extrinsic are the ones in which the origin are beyond the palm, whereas intrinsic muscles are present within the hand. Extrinsic muscles are essentially long flexors and extensors. They are usually the bulk of the muscle is usually located in the forearm, where with tendons projecting towards the hand uh, through uh, through the wrist. Examples of extrinsic muscles are flexor digitorum superficialis, profundus, extensor digitorum communis, etc. Internal intrinsic uh, muscles include thenar, hypothenar, interossi, and lumbricals. These are muscles which are very small, and these are located within the hand itself. So, enslaving is an involuntary force production by non-intended fingers. The intended finger might be one where uh, the uh, the finger for which the movement is intended, say for the index finger, uh, then the index finger is termed as the master finger, whereas other force producing fingers for which the movement was not intended are known as slave fingers. Due to this enslaving, there is no direct correspondence that includes that exists between the neural commands to the individual fingers and the forces produced by these fingers. um, and therefore the scaling should be in accordance to the should take into account the command sent by mistakenly sent into the other fingers. Some of the hypothesized reasons are uh, peripheral mechanical coupling between the tendons, which leads to various shear and varying shear between the tendons, and therefore the forces are transmitted to other phalanges as well. So essentially enslaving force is a quantitative measure. It can be because of various factors. The first one is peripheral mechanical coupling between the tendons, which lead to intertendinous interactions and therefore control of other uh, movement in other digits as well. Multi-digit motor units in extrinsic flexor and extension muscles, uh, and diverging central commands. Coming to the first one, which is peripheral mechanical coupling between the tendons, these are frequent. The tendons within the hand are frequently connected to each other in by fascia-like structures, and because of these connections, uh, there are there might be finger enslavement happening. However, this is not true for uh, the this is to some extent uh, done away by various pianists, players, musicians, which need a very high dexterity in the hand. uh, However, things that require very independent finger movements can give rise to some difficulties for even very experienced musicians. Coming to the second reason, which is that which is multi-digit motor units in the extrinsic flexor and extension muscles. If you What does muscle motor unit mean? Motor unit is essentially the neuron that arises from the spinal cord and goes to supply into the region of interest. So it is uh, essentially actual group of muscle fibers in a muscle innervated by a single neuron which arises from the spinal cord. And so every skeletal muscle fiber is innervated by an axon that is connected to a neuron which arises from the spinal cord in order to contract. So the command to contract comes from the spinal cord, traverses through the body of the neuron, and then finally there is neurotransmission occurring at the junction at the synapse. The assumption is that each muscle fiber is innervated by only one motor neuron. However, this might not be true. There might be overlaps between the motor units, and uh, because of these, the muscle fibers from these the enslavement might arise because as a result of divergence of the muscle fibers from these particular motor units. And there might be commonalities between the motor units, and therefore this can lead to enslavement. One more uh, hypothesis is the divergence of central commands. So a large research area is dedicated to understanding the organization in the cortex, and which area leads to uh, what kind of uh, each each area within the brain has, or the within the motor cortex has controls a particular single site. And the hypothesis is that the primary motor cortex has a tiny human inside it, with each area supplying nerves to a particular area, and therefore controlling that area. And there is a neurological map of the areas, and the which are dedicated to processing motor functions, sensory functions for different parts of the body. Each area is responsible for one single part. However, this is the old view. However, the new view suggests that these cortical representations might overlap. While there are the neuronal supplies might be different for index, for the middle finger, for the ring finger, and for the uh, little finger. The In the motor cortex, they might arise from a single area, and these cortical representations therefore uh, overlap. Because they overlap, they there is bound to be mismatch of the mix up in the signals, and therefore enslaving might occur. And this has been confirmed using some neuron recordings in monkeys, that the cortical representations overlap in uh, for different fingers. Okay. during um, During joint movement, the tendons are slide over some distance, and therefore they must overcome the gliding resistance, which is the shear that they'll feel by the environment. Usually, these tendons are surrounded by a sheath which provides them nutrition, which protects them from friction. One of those A lot of the joints within the human body are synovial fluid on them in them. The synovial fluid provides the lubricant of sorts, which keeps the friction at low values. However, some of these tendons will might not have synovial fluids, and these are known as extra synovial tendons. uh, Gliding resistance in itself depends on many factors. One is lubrication, relative size of the tendon. Larger the tendon, larger the resistance, and the surrounding sheath. The frictional coefficient between the flexor tendon, which is this one, and A2 pulley. So there are different pulleys that connect, um, that help in connection of the tendon to a particular bone. The it's for flexor tendon and A2 pulley, the frictional coefficient has been measured to be between 0.02 to 0.063. In case of an injury, there is a reduction in gliding resistance, which there is an increase in the gliding resistance. And to reduce the gliding resistance uh, after the repair surgery, there needs to be a proper suturing, proper biomaterial, proper lubrication provided. Otherwise, there will be a very high gliding resistance, and that will affect the movement of the tendon. So, tendons can be of two types. They can either be direct, they connect to the bone without wrapping, or they can be wraparound tendons, which that bend around a pulley. And in this case, this is the pulley. When a curved tendon exerts a force, they also exert a force towards the center of the curvature and then straighten out. Uh, And when such wraparound tendon slide, they feel a very high frictional force as compared to direct tendons. And for modeling assumptions, it's assumed that uh, wraparound tendons are wrapped around a cylindrical structure, say for example bone or another muscle. Let's look at the analysis for this. Say a tendon wraps around a pulley, and for this particular analysis, T2 is greater than T1, and the movement is in this direction. The angle of contact is beta, uh, which is the angle of lap or the angle of contact. R is the radius of the cylinder. T1 is the slack side belt tension, and T2 is the tight side belt tension, which is higher. And if you look at a very small area, okay, let me just go to, if you look at a very small area, you will see that this is the let this be the tangent to that area, and let the point of angle of contact be d theta. So the tension that is on this side will be T, the tension on this side will be T plus dt, right? And the angle that this forms is theta by d theta by 2, and similarly d theta by 2. Because of the contact, there will be a normal force produced. The normal force is dn, and basic frictional principles, the friction will act normal to the force produced. If this is the normal force, friction will act in this direction, and because the movement is occurring in this direction. Okay, you can either consider this as the xy, or you can consider this as the normal and the tangential directions. So if you look at normal and tangential directions, in the normal direction, what forces do you have? You have dn, you have the you have these components of the tension. So this will be t plus dt sine, one second, this will be t plus dt sine d theta by 2, and this will be t plus dt cos d theta by 2. Similarly, this will be t sine d theta by 2, and this will be t cos d theta by 2. So in the normal directions, the forces are dn and the sine components of t plus dt and t. So dn is in the positive direction because minus t sine theta d theta by 2 minus t plus dt sine theta d theta by 2 is equal to 0 because the force is, there is no external force applying on it. Now dn can be written as t sine d theta by 2 plus t plus dt sine d theta by 2. For very small angles, if theta is very, if d theta is very small, sine d theta by 2 is equal to d theta by 2. Cool. So dn can be written as t d theta by 2 plus t d theta by 2 plus dt d theta by 2. This becomes 0 because there are two differentials, so very small quantity. So dn can be written as t d theta. Okay. For the tangential direction, so if you were to look at the tangential direction, what forces are there? The frictional component, and the cos components of both of these tensions. So let's write that down. T d cos d theta by 2 plus mu dn, the frictional force, minus t plus dt t cos theta by 2 in the positive direction, dn in the positive direction, t plus dt cos theta by 2 in the cos d theta by 2 in the minus direction, cos d theta by 2. If you solve this, for d theta tending to 0, very small d theta, cos d theta by 2 will tend to, essentially if theta is very close to 0, cos theta tends to 1. Okay, so mu dn can be written as dt because cos d theta by 2 is 1. So we got one equation here and one equation here. If I substitute 1, substitute 1 and 2, so I know dn is t d theta. I can write 2 as mu times t d theta is equal to dt. Mu d theta is equal to dt by t. If you apply integral on both the sides, so theta will range from 0 to beta, and tension ranges from T1 to T2. Mu is constant, so it essentially becomes mu times theta 0 to beta. So t is 1 over t will be logarithm of ln t. So mu times beta is equal to L and T2 by T1 into the T ln T2 minus ln T1, which can be further written as ln T2 by T1. Applying exponential, if you take it to power, T2 by T1 becomes E times mu beta. T2 can be written as T1 E times mu beta. Okay, so this is the relation between the tensions that are felt by both the sides. Okay, so this is known as Euler Einstein formula or Capstan equation. You can further denote this in this formula where Fp is the force at the proximal end of the tendon and Ft is the force at the distal end of the tendon. So the frictional force finally will be this value. Fd, Fp, F proximal minus F distal, which will result in a formulation like this. So usually the measurements for frictional values can be done by dissecting tendons and pulley from cadavers. In this scenario, tendon and pulley, flexor digitorum profundus tendon and A2 pulley were dissected out, and then a load of 4.9 Newton was applied on one end, resistant to a linear actuator on the other end. This is the load, this is the actuator connected by a nylon rope, and there were sensors to measure forces F1 and F2. And the angles were varied. It was observed that depending on the angle of the angle of alpha and beta, the frictional force varied from 0.021 to 0.031 Newton. And therefore, from using this, the coefficient of friction was calculated to be between 0.022 to 0.063, which is very small. Okay, that is the end of the summary for week 9. I will just present the screen. Okay, so let's look at some questions pertaining to week 9. The first one is the negative electrostatic charge on proteoglycans help in binding of sugars, covalent bonds forming between two GAG molecules, binding keratin sulfate chain, and water retention. The answer is water retention. We saw this during the classes. The structural hierarchy in tendons are which of the following? Fascicles, fibril, subfibril, microfibril, and tropocollagen is the answer. Elastic behavior of tendons is described by tendon stiffness, Young's modulus, compliance, all of the above. All of these terms describe the elastic behavior of tendons. In low strain conditions, in the toe region of the stress strain relation of a tendon, slope is maximum, slope is linear, Uh, Collagen fibers straighten and lose their crimp, microscopic failure of fiber bundles occur. The answer is collagen fibers straighten and uh, lose their crimp. In the first model of nonlinear elasticity in the toe region, which is the primary assumption regarding collagen fibrils, they remain static throughout deformation. They are uniformly recruited at maximum crimp. They are recruited consecutively at varying degrees of crimp. They are pre-stressed before deformation. The answer is they are recruited consecutively at varying degrees of crimp. And at when they enter the linear region, the assumption is that all of these have straightened out. All of the fibers have straightened out. How does the flexible hinge model compare the straightening of crimped collagen fibers to a familiar mechanical analogy? Uncoiling of a spring spiral, compression of a spring, extension of a rubber band, stretching of a helical spring until it becomes a straight wire. The answer is stretching of a helical spring until it becomes a straight wire. Which of the following statements is true? One third of the protein in the body is collagen. Elastin is most common constituent of ligaments followed by collagen. Collagen is the most common constituent of tendons followed by elastin. Collagen has a double helical structure. The answer is one third of the protein is collagen. Elastin is uh, collagen is actually the most common constituents in both the tendon and ligaments, and tropocollagen has a triple helical structure, so the last statement is wrong. The structure of the collagen is characterized as a double helix, triple helix, crimped fibers, presence of glycosaminoglycan. The answer is the triple helix. Which of the following types of cartilage is found in synovial joints? Hyaline, elastin, proteoglycan, fibrocartilage. The answer is hyaline. Fibrocartilage is uh, found in intervertebral discs. Select all the true statements. Collagen fibers in tendon are more parallel than in ligaments. Collagen fibers in ligaments are more parallel than in tendons. Tendon and ligaments have hierarchical arrangement of fibers. Tendons have hierarchical organization of collagen fibers, whereas in ligaments, all the collagen fibers are bundled together. The answer is collagen fibers in tendons are more parallel than in ligaments, and tendons and ligaments have hierarchical arrangement of fibers. Okay. That's it for week 9. Thank you for listening.